Hello students, I am Grishma Dalal from Faculty of Pharmacy, Dr. Subhash Technical Campus. This is the continuous portion of the last video lecture that I prepared, that is Pharmaceutical Engineering Centrifugation 1.3. In this video, we will see about some of the centrifuges, right? If you remember, when I prepared centrifugation 1.1, at that time we studied the definition of what do you mean by centrifuge. Centrifuge are the equipment that are used to separate different solutions or different mixtures, you can tell. So we are going to see that how centrifuges work. So these are the classification based on the mechanism of separation. Here we are mainly going to focus on mechanism. So please focus here. The first is sedimentation centrifuge. Difference in the densities of two or more phases of mixtures. If there is a difference in density, which you easily understand now, then it is called sedimentation centrifuge. Ultracentrifugation is a part of sedimentation centrifuge. Here in ultracentrifugation, you just try to get more pure separation. Then if you use simple centrifugation processes or simple centrifuges, you get more pure in ultracentrifugation method when extremely fine solid matter is needed to be separated. What are the uses of these both? First is suspension containing low concentration of solid can be separated quickly. Lanolin is recovered from the wool by scouring process. We will discuss about this process deeply. Olive and fish liver oils are purified by removing dirt and water. The liquid phases are separated in extraction of drugs, for example, manufacturing of antibiotics. So these are the uses in which sedimentation type of centrifugation is used heavily, and in some of the cases you can also use ultracentrifugation technique too. Bacterial enzymes are also prepared by removing the bacteria. Blood plasma is separated from the whole blood. Starch is collected after washing and purification stages. In the manufacture of insulin, liquor is clarified so as to remove the precipitated protein. Now, before we move further, do you know what is clarified means? Right, clarified, we have studied in filtration that it is a type of a clarification, is a type of a filtration, right? Which means in manufacture of insulin, liquor is filtered or separated. You can easily call it is a type of a separation so as to remove some of the precipitated protein that are not required, or some are most required. Advantages of these type of centrifuges that work on sedimentation principle is centrifuges are compact and occupy a very small space. The centrifuges which are solely depend on the mechanism of sedimentation have really mini size, so you can fit in your lab too. The separating efficiency is really high so that the particles are deposited very rapidly. It is suitable if the sediment is a fluid deposit or a sludge. The separating efficiency in this case, you can really connect it because ultracentrifugation is really important in separating very minute, even minute size of globule size or particle size. Then ultracentrifugation will be really helpful in separation. Two immiscible liquids are easily separated on a continuous process. By controlling speed of rotation and rate of flow, coarse particles are separated while finer particles remain in the suspension. By controlling the speed of rotation and rate of flow, coarse particles are separated. When you control the speed of rotation and rate of flow, you can easily separate the particle size that are a little bit big in size, while finer particles remain in the suspension. Now, obviously there must be disadvantage. So the first is the construction of sedimentation centrifuge is complicated. Second, its capacity is limited. These are the most important disadvantages which any person who selects the centrifuge have to keep in mind that the capacity is limited. Now, second is filtration. The first one we saw was sedimentation, but the second one is filtration centrifuge. Filtration centrifuge is a centrifuge in which solid parts are retained by porous medium, allowing liquid to rush out due to centrifugal effect. In this type of container, contains a porous wall through which the liquid phase may pass and on which solid phase is retained. Example are perforated basket centrifuge and semi-continuous centrifuge. Can you imagine any practical example which uses filtration centrifuge? See, I'm not recommending any practical examples here, but this type of filtration you can easily compare in your practical example, like whenever somebody is filtering a tea, right? This is this type of centrifuge. The only difference here, it uses centrifugal force or centrifugal effect to separate. There will be a cloth or any porous medium on which the solid particles will be retained. Retained means they will be captured, that solid cannot pass through, right? You must have also performed the filtration practicals or experiments in your lab scale, right? So this depends on, this is a really simple principle. This is a really simple centrifuge, and examples are perforated basket centrifuge and semi-continuous centrifuge. Uses: when solid recovery is the primary goal, when it is the only sole goal, centrifugation must be considered as an alternative to simple filtration. As I spoke, that simple filtration, it is a, you can say it is a variant, right? You can connect a centrifuge with simple filtration technique and separate particles. It is also used for removing unwanted solid from a liquid. For example, precipitated protein are removed from insulin. It is really important, and it is really useful in removing the solids that are not needed in any liquid, right? Advantages: it can handle slurries with high proportion of solid and even those having paste like consistency. Can you just remember what the advantage, sorry, what the disadvantage was in the past sedimentation centrifuge? That was that it didn't handle the high proportion of the slurries. It was capacity limited. So here it can handle slurries which have high proportion of solid and even those having high paste like consistency. The final product has very low moisture content. Here moisture content is very low when you get the final product. In this method, the dissolved solids are also separated from the cake. Where if the cake is formed, then the dissolved solids are separated in via filtration centrifugation. Disadvantages: the entire cycle is complicated, resulting in considerable labor cost. So economically, it is a disadvantage. It is a batch process or a semi-continuous. You cannot keep it a fully continuous process, right? You have to work in batches, or you can have a semi-continuous type. The solid may form a hard cake due to centrifugal force, which is difficult to remove. If the hard cake forms, then you have to take measures to remove it, which is a tedious job. So that's it. This is all about centrifugation, and here the final centrifugation I will complete. In our next video lecture, we will be focusing on materials of plant constructions. So stay tuned. Thank you for your patience. Thank you.